Hello, my name is Kristina Dahlgren and I'm a graduate student in the Hammond Cognitive Neuroscience Laboratory at Emory University. And today I'm going to talk to you about my poster, The Neural Correlates of Emotional Episodic Memory, Encoding and Retrieval. This is a neuroimaging meta-analysis using seed-based demapping. Emotional events are remembered in greater detail with more accuracy and they last longer. A lot of different labs have used a variety of different tasks and stimuli to study emotional memory using functional magnetic resonance imaging, and they all end up getting slightly different results. Quantitative meta-analytic methods allow us to examine these fMRI results across the different tasks and labs to determine what brain regions are consistently activated across studies. In 2010, Murdy et al. Conducted a quantitative meta-analysis examining the brain regions that were active during encoding of emotional items that were later remembered. Since then, the methods used to conduct quantitative meta-analyses have improved substantially, and there has been 10 more years of research into emotional memory. In addition, a more recent focus of emotional memory has been in identifying the brain regions that are active during retrieval of emotional information. Here, we conducted an updated meta-analysis of neuroimaging studies that examined emotional memory during encoding and a brand new meta-analysis of studies that looked at emotional memory during retrieval. Finally, we identified brain regions that were active during both encoding and retrieval. We predicted that some of the findings from the previous 2010 meta-analysis would be replicated here, such as the amygdala and hippocampus. However, we also predicted that due to the larger data set and the new meta-analytic method, we might find that some of the brain regions that were identified in the previous data set would no longer be active, and that we would identify some new regions that were not previously identified. To conduct our meta-analysis, we first ran a search in PubMed using search terms with various synonyms for emotion, memory, fMRI, and either encoding or retrieval. In addition, we also used review articles, reverse citation searches, and a Google Scholar search to identify possible articles. We ended up with 526 papers for our encoding analysis and 419 papers for our retrieval analysis. Of those papers identified in the searches, we ended up with 25 papers for encoding and 17 papers for retrieval that met our inclusion criteria. If you're interested in additional details regarding the included papers, I have attached the tables of included papers as an additional file in the virtual booth. For our statistical analysis, we use the seed-based demapping method with permutation of subject images. To correct for multiple comparisons, we use threshold-free cluster enhancement with family-wise error correction set to p less than 0.05. We performed 1,000 permutations. For the results of our encoding meta-analysis, we found that successful emotional memory encoding recruited brain regions bilaterally in the medial temporal lobe that are commonly associated with episodic memory encoding, including the hippocampus, entorhinal cortex, perirhinal cortex, and the parahippocampal cortex. We also found activation in brain regions that are involved in emotional processing, including activation bilaterally in the amygdala, orbitofrontal cortex, temporal pole, as well as in the left putamen and the insula. And these regions, especially the insula, are commonly associated with empathy processing. We also found bilateral activations in brain regions involved in perceptual processing, more specifically visual processing, including the lateral occipital cortex, fusiform cortex, and the inferior and middle temporal gyri. For emotional memory retrieval, we also found activations in some of the same medial temporal lobe regions that have consistently been shown to be critical for both episodic memory encoding and retrieval, including bilateral activations in the hippocampus, entorhinal cortex, and the perirhinal cortex. We also found regions outside the medial temporal lobe that have been implicated in episodic memory retrieval, including bilateral dorsolateral prefrontal cortex and activation in the left angular gyrus. We also found regions that have previously been implicated in emotional processing, including bilateral activations in the amygdala And we also found activations in the left hemisphere in the orbital frontal cortex, temporal pole, putamen, and insula. We also found activation in regions involved in perceptual processing, including the bilateral occipital cortex, the middle occipital cortex, the middle temporal gyrus, and Heschel's gyrus. Finally, we looked at brain regions that were identified as being consistently activated across studies of both emotional memory encoding and retrieval. As you might expect, based on the findings for encoding and retrieval, we found areas of overlapping activation in the medial temporal lobe regions associated with both episodic memory encoding and retrieval, including the hippocampus, entorhinal cortex, perirhinal cortex. However, these medial temporal lobe activations were only observed in the left hemisphere. For regions that are associated with emotion processing, we observed activation in the amygdala bilaterally and activations in the left orbitofrontal cortex, temporal pole, putamen, and insula. We also observed activation in bilateral perceptual regions, including the lateral occipital cortex. In conclusion, successful emotional memory encoding recruited brain regions that are associated with episodic memory encoding, emotional processing, and perceptual processing. Successful emotional memory retrieval recruited many of the same brain regions associated with emotional memory encoding, including regions associated with episodic memory encoding, emotional processing, and perceptual processing. Additionally, emotional memory retrieval also recruited additional brain regions associated with episodic memory retrieval that were not identified during emotional memory encoding, including the dorsolateral prefrontal cortex and the angular gyrus. Compared to the previous emotional memory encoding meta-analysis, many of the same regions were activated in our analysis, including the amygdala and the medial temporal lobe memory system. However, some regions identified in the previous meta-analysis did not persist in the current meta-analysis, including activations in the prefrontal and parietal cortex. Finally, we observed activations in areas that were not previously identified in the Murdy et al. Meta-analysis, including the insula. Thank you for coming to my poster virtually. If you would like additional information, you can email me at Kristina with a K, .dahlgren@emory.edu. You can also visit the lab website, hammond.weebly.com. And also keep an eye out for this publication that will be coming out soon in the Tolving special issue of Neuropsychologia. If you'd like additional information on when that will be published, or if you would like to obtain a copy, again, please email me. And feel free to ask questions in the chat function. Thank you.